Hello everyone, let's now read the mediators of inflammation. These are large and increasing number of endogenous chemical substances which mediate the process of acute inflammation. Endogenous substances. Mediators of inflammation have some common properties as under. These mediators are released either from the cell or are derived from plasma protein. Cell-derived mediators are released either from their storage in the cell granules or are synthesized in the cell. The most common site of synthesis of plasma-derived mediator is the liver. After their release from the liver, these mediators require activation. All mediators are released in response to certain stimuli. These stimuli may be variety of injurious agent, dead and damaged tissue, or even a mediator stimulating release of another. The latter are called secondary mediators, which may perform the function of initial mediator or may have opposing action. Mediator acts on different targets. They may have similar action on different target cell or differ in their action on different target cell. They may act on cells which form them or on the other body cells. Fourth, range of action of different mediators are increased vascular permeability, vasodilation, chemotaxis, fever, pain, and tissue damage. Mediators have short lifespan after the release. After the release, they are rapidly removed from the body by various mechanisms, for example by enzymatic inactivation, antioxidants, regulatory proteins, or even decay spontaneously. Two main groups of substances acting as chemical mediators of inflammation are those released from cell and those from the plasma protein. Mediators of inflammation. Cell-derived vasoactive amines like histamine, 5-hydroxytryptamine, neuropeptides, arachidonic acid metabolites like eicosanoids, metabolites via cyclooxygenase pathway like prostaglandin, thromboxane A2, prostacyclin, resolvin, and metabolites via lipoxygenase pathway like 5-HETE, leukotrienes, and lipoxins. Third, lysosomal component from PMNs and macrophages, platelet activating factors, cytokines like interleukin 1, 6, 8, 12, IL 17, tissue necrotic factor alpha, beta, IFN, gamma, and chemokines. Free radical oxygen metabolite nitric oxide. Plasma protein derived mediators. Plasma proteases products of kinase system, clotting system, fibrinolytic system, and the complement system. The range of actions in acute inflammation are shown in figure 5.7. Figure 5.7 Mediators of inflammation. Cell derived are derived from the source, mast cell, basophils, platelets, is histamine, which main action is increase in the permeability. Platelets mediator is serotonin, which also increases the permeability of the vessel. An inflammatory cell, for example prostaglandins, leukotrienes, lysosomal enzymes, platelet activating factor, cytokines, and nitric oxide and oxygen metabolites. These all so prostaglandin will cause vasodilation, leukotriene will lead to increase in the permeability, lysosomal enzyme will cause tissue damage. Platelet activating factor will increase the permeability. Cytokine will cause fever, and nitric oxide and oxygen metabolite will lead to tissue damage. Plasma protein derived clotting and fibrinolytic system. Fibrin split the product and increase the permeability. Kinin system like kinin or bradykinin increase the permeability, and complement system anaphylactones like C3A, C4A, C5A, C5B and C9 will also lead to increase in the permeability. Now let's see the cell-derived mediators. So cell-derived mediators, first is vasoactive amine. Two important pharmacological active amine that have role in the early inflammatory response. First one are histamine and 5-hydroxytryptamine or serotonin. Another addition to this group is neuropeptide. First, histamine. It is stored in the granules of mast cells, basophils and platelets. Histamine is released from these cells by various agents as under stimuli or substances including acute inflammation, for example heat, cold, irradiation, trauma, irritant, chemical, immunologic reactions. Then second is anaphylactones, like fragments of complement C3A and C5A, which increase the vascular permeability and causes edema in the tissue. Histamine releasing factors from neutrophils, monocytes and platelets. Leukotrienes. The main action of histamine are vasodilation, increased vascular permeability, itching and pain. Stimulation of mast cell and basophil are also release the product of arachidonic acid metabolism, including the release of slow reacting substances of anaphylaxis, SARS-AS. The SARS-AS consists of various leukotrienes like LTC4, LTD4 and LTE4. Second is 5-hydroxytryptamine or serotonin. It is present in the tissues like chromaffin cells of GIT, spleen, nervous tissue, mast cell and platelets. The action of 5-hydroxytryptamine are similar to histamine, but it is a less potent mediator of increased vascular permeability and vasodilation than histamine. They may be mentioned here as carcinoid tumor in the serotonin secreting tumor. Third is neuropeptide. Another class of vasoactive amine is tachykinin. Neuropeptides such as substance P, neurokinin A, vasoactive intestinal peptide and somatostatin. These small peptides are produced in the central and peripheral nervous system. The major pro-inflammatory actions of the neuropeptide are as follows: increased vascular permeability, transmission of pain and mast cell degranulation. So they increase the permeability, causes the stimuli, transmission of pain and degranulation of mast cell. So first cell mediator was amines which are vasoactive, which include histamine, serotonin and neuropeptide. Now let's see arachidonic acid metabolites, eicosanoids. Arachidonic acid metabolites or eicosanoids are the most potent mediator of inflammation, much more than oxygen free radical. The arachidonic acid is a fatty acid eicostate Eicosapentaenoic acid. Greek word eicosa means 20 because of 20 carbon atoms composition in this fatty acid. Arachidonic acid is the constituent of phospholipid cell membrane. Besides its presence, some constituent in diet, arachidonic acid is released from the cell membrane of phospholipid. It is then activated from arachidonic acid metabolites or eicosanoids by one of the following two pathways: cyclooxygenase pathway or lipooxygenase pathway. First metabolite via cyclooxygenase pathway, prostaglandin, thromboxane A2 and prostacyclin. The name prostaglandin was first given to a substance found in human semi-fluid, but now the same substance the name prostaglandin was first given to a substance which was found in human seminal fluid, means the semen of which includes sperm. But now the same substance has been isolated from a number of other body cells. Prostaglandins and related compounds are also called as autocoids because these substances are mainly autocrine or paracrine agents. Means these substances are produced by the cells which need their action or the cells which are present in the nearby to the cell which needs them. The terminology used for prostaglandin is abbreviation as PG, followed by the suffix of an alphabet and a serial number, example PGG2, PGE2, etc. Cyclooxygenase COX, a fatty acid enzyme present as COX1 and COX2, acts on activated arachidonic acid to form prostaglandin endoperoxide PGG2. PGG2 is enzymatically transformed into PGH2 with generation of free radical of oxygen. PGH2 is further acted upon by enzymes and result in the formation of the following three metabolites: prostaglandins D2, E2 and F2 alpha. PGD2 and E2 acts on blood vessel and cause increase in the venular permeability, vasodilation and bronchodilation and inhibit the inflammatory cell function. PGF2 alpha induces vasodilation and bronchoconstriction. Thromboxane A2 TXA2 platelets contain the enzyme thromboxane synthase and hence the metabolite thromboxane A2 formed in active in platelet aggregating beside its role as vasoconstrictor and bronchoconstrictor. Prostacyclin PGI2 induces vasodilation, bronchodilation and inhibits platelet aggregation. Resolvins are another derivative of COX pathway which act by inhibiting the production of pro-inflammatory cytokines. Thus, resolvins are actually helpful drugs such as aspirin act by inhibiting COX activity and stimulate the production of resolvin. Resolvin is kind of tricky. Resolvin is another derivative of COX pathway which act by inhibiting the production of pro-inflammatory cytokines which leads to inflammation that are inhibited by resolvin. Those are actually helpful drugs such as aspirin acts by inhibiting the COX activity and stimulate the production of resolvin. They may be mentioned here that some of the major anti-inflammatory agents act by inhibiting the activity of enzyme COX. For example, non-steroidal anti-inflammatory drugs and COX-2 inhibitors. Second is metabolism via lipooxygenase pathway, 5-HETE, leukotrienes and lipoxins. The enzyme lipooxygenase, a predominant enzyme present in the neutrophils, acts on activated arachidonic acid to form hydroperoxyecostetraenoic acid, 5-HPETE, which on further peroxidation forms the following two metabolites, 5-HETE hydroxy compound and intermediate product is a potent chemotactic agent for neutrophils. So in the lipoxygenase pathway, enzyme is neutrophils are activated arachidonic acid to form the 5-HPETE, which then there occurs the peroxidation of 5-HPETE. So the compound thus formed first is 5-HETE, which is an intermediate product, is a potent chemotactic agent for neutrophils. Means the neutrophils will get attracted towards the site where this compound will be present. And second is leukotrienes, are so named as they were the first isolated from leukocytes. Firstly, unstable leukotriene A4 is formed, which is acted upon by the enzyme to form LTB4. Chemotactic for phagocytic cells and stimulate phagocytosis. They are chemotactic for phagocytic cell and will stimulate phagocytic cell adherence. Cells like LTC4, D4, E4 have common action by causing smooth muscle contraction and thereby induces the vasoconstriction, bronchoconstriction and increased vascular permeability. So the two main factors which leads to acute inflammation are vascular permeability and vasodilation. It is often asked by tutors in Viva. So where were we? Bronchoconstriction and increased vascular permeability. Hence they are called as slow reacting substance of anaphylaxis, SARS-AS. Third is lipoxin, LX, act to regulate and counterbalance the action of leukotrienes. Lipoxygenase 12, they regulate and counterbalance the action of leukotrienes. What was leukotriene causing? It was causing vasoconstriction, bronchoconstriction and increased vascular permeability. So reverse will be done by lipoxins, which are present in platelets and acts on LTA4, which is derived from neutrophil, and it will form LXA4 and LXB4. Third is lysosomal components. The inflammatory cells, neutrophil and monocyte contain lysosomal granules, which on release elaborate a variety of mediator of inflammation. These are as under. First is granules of neutrophil. Neutrophils have three types of granules: primary or azurophil, secondary or specific, and tertiary. First is the primary or azurophilic granules are large azurophil granules which contain functionally active enzyme. These are myeloperoxidase, acid hydrolases, acid phosphatase, lysozyme, defensin, cationic protein, phospholipase, cathepsin G, elastase and protease. Then the secondary or specific granules contains alkaline phosphatase, lactoferrin, gelatinase, collagenase, lysozyme, vitamin B12, binding protein, plasminogen activator. Let's read it again because they are so much. So primary or azurophil granules are large azurophil granules which contain functionally active enzymes. These are myoperoxidase, acid hydrolases, acid phosphatases, lysozyme defensin, cationic protein, phospholipase, cathepsin G, elastase and protease. Whereas secondary or specific granules contain alkaline phosphatase, lactoferrin, gelatinase, collagenase, lysozyme, vitamin B12 binding protein and plasminogen activator. Activated arachidonic acid is acted upon by lipoxygenase which lead to formation of 5-HPETE which is chemotactic, and then it will be degraded to leukotriene A4 which will be further degraded into LTB4 which is again chemotactic and cell adherence, LTC4, D4, E4 and lipoxins. All function as smooth muscle constrictors, vasoconstrictor, bronchoconstrictor and increase vascular permeability. Arachidonic acid metabolites via lipoxygenase pathway. Now let us see what are tertiary granules or C particles contain gelatinase and acid hydrolase. Myeloperoxidase cause oxidation lysis by generation of oxygen-free radicals. They cause oxidative lysis by generation of oxygen-free radical. Acid hydrolysis acts within the cell to cause destruction of bacteria into phagolysosomes, while proteases attack on the extracellular constituents such as basement membrane, collagen, elastin, cartilage, etc. However, the degradation of extracellular component like collagen, basement membrane, fibrin and cartilage by protease result in harmful tissue destruction which is kept in check by presence of anti-protease like alpha-1 antitrypsin and alpha-2 macroglobulin. So tertiary granules or C particle contain gelatinase and acid hydrolases. Myeloperoxidase cause oxidative lysis by generation of oxygen-free radical. Acid hydrolase act within the cell to cause destruction of bacteria in phagolysosome, which protease attack on extracellular component such as basement membrane, collagen, elastin, cartilage, etc. However, degradation of extracellular component like collagen, basement membrane, fibrin, cartilage by protease results in harmful tissue destruction which is kept in check by presence of anti-proteases like alpha-1 antitrypsin and alpha-2 macroglobulin. Second are the granules of monocytes and tissue macrophages. These cells on degradation also release mediators of inflammation like acid protease, collagenase, elastase, plasminogen activator. However, they are more active in chronic inflammation than acting on mediators of acute inflammation. Granules of monocyte and tissue macrophage. These cells on degranulation release mediators of inflammation like acid proteases, collagenase, elastase, plasminogen activator. However, they are more active in chronic inflammation than acting on mediators of acute inflammation. Platelet activating factor. Platelet activating factor is released from PGE sensitized basophil or mast cells, other leukocytes, endothelium, and platelets. However, from its action on platelet aggregation and release reaction, the action of PAF on mediators of inflammations are increased vascular permeability, vasodilation, and low concentration and vasoconstriction, otherwise bronchoconstriction, adhesion of leukocyte to endothelium, and chemotaxis. We will continue again from platelet activating factors.